Hi, this slide shows the main problem addressed in this study. Bone defects can be caused by trauma, disease, or congenital conditions. Test defects result in a loss of bone structure and fusion. In many cases, bone cannot heal properly on its own. The current gold standard treatment is use and autograft. However, this method has important limitations. Therefore, there is not developed and advances the biomaterials for bone regeneration. Okay, and this slide explains the justification of the study. Bone defects require effective strategies for regeneration because natural healing is limited. Current treatments, such as autograft, present important limitations, including limited availability and complication. Okay, for this reason, tissue engineering has emerged as an alternative approach. In this field, biodegradable scallops are used to support cell growth and tissue regeneration. In addition, bioactive coaching can be incorporated to improve cell adhesion and stimulate bone formation. Therefore, the goal of the study is to develop an scaffold with bioactive properties to enhance bone regeneration. Okay. Okay, this slide presents the material used in the study. The main material is biodegradable polymer KPCLTMA, which used to fabricate the scallop structure. The material is important because it can degrade over time and place beneath bone tissue. The scaffold is produced using 3D printing technology, specifically stereography, which allows precise control of the structure. Okay, this is important to mimic the architecture of natural bones. This is very important. Okay, in addition, the scaffold surface is modified using different bioactive coatings. These include elastin-like polypeptides known as LP, which improve biocompatibility. Another coating is a combination of polyethylene acrylate, fibroconnectin, and BPM2, which are to use it and cellulation and stimulate bone formation. Um, to evaluate biocalic response, human bone marrow stromal cells are used since they can differentiate into bone cells. Finally, in vivo models such as the CAM assay and mouse model are used to angiogenesis and bone formation in more realist environments. This slide presents the method used in the study. The method includes both in vitro and vivo experiments, as well as material characterization techniques. In vitro analysis was performed to evaluate cell behavior of the scaffold. Cell viability was measured using VLR view, which indicate how well cells survive and grow. In addition, in addition, alkaline phosphate activity was evaluated to determine osteogenic differentiation. Sorry. Gain expression analysis was all perfect using PCR study the expression of bone relative genes. For in vivo analysis, the CAN assay was used to evaluate angiogenesis and biocompatibility. The test allows observation of blood vessel formation around to the scaffold. Additionally, a mouse model was used to analyze bone formation in a more realistic biological environment. Okay, finally, the material was characterized using different techniques. Scanning electron microscopy was used to observe the scaffold structure. Ferriti spectroscopy was to analyze the chemical composition, okay, present. And mechanical testing was perfect to evaluate the strength and stability of the scaffold. Okay, continuation. Okay, thank you.
In the results, the figure 1 shows the complete characterization of the PCL and TMIA scaffold material. This structure is porous and interconnected, and the surface is not completely smooth because actually promotes the cell adhesion because it increases surface roughness. And finally, the geometry is reproducible thanks to stereolithography printing. In the panel C show the FTR analysis of the printed scaffold, which allow us to identify the chemical groups on the material surface. The dominant peak responds to the carbonyl group, characteristics of the ester bonds present in the polycarbonate chains. These confirm the material chemical identity. The most important point is the spectrum in the carbonyl group is present is small but detectable, indicating that very few metrical groups. And finally, this confirms that the degree of functionalization was greater than 95%, which tells us that the material polymerized almost completely, giving the desired mechanical properties. In the panel D shows the compression curve of the scaffolds. The blue line represents a different scaffold, and the similarity of these curves indicates the good mechanical reproducibility. The range of cancelable bonds between 100, 5, and 500 megapascals, so our scaffold falls within the lower range of that tissue. Furthermore, the scaffold withstood strains up to 15 to 20% before failure, which is approximately twice the maximum strain of cancellous bone, indicating that the material is more ductile than a natural bone. And remaining in the panel E shows the celebrated degradation study of the material. It utilizes sodium hydroxide at 37 degrees to accelerate the degradation condition that, for example, in the 7th day onward, a trend toward mass loss becomes noticeable, and in the day 21, the scaffold have lost approximately 50 to 20% of the original mass. And the smaller bar bars indicate that the tree scaffold behaves consistently, and this result obviously is crucial because it confirms that PCL antimia is biodegradable. Okay, this slide shows the in vitro results for the cell viability and the donation of the PCL and TMEA scaffolds with the different bioactive coatings. First, in the panel B, we can see fluorescence microscope image of the human bones, stroma cells, and the scaffolds of the two time points, the day 1 and the day 14. The four columns represent the four conditions tested. Obviously, in the gray color, right, represents the live cells, and the red color represents the dead cells. And now in the panel A on the right, this bar graphic shows the alarma blue fluorescence results, which is surrogate for the cell number, metabolic activity. On the X, for example, in the X-axis, we have the two times point of day 1 and the day 14, and the G-axis, we have fluorescence in arbitrary units. They was finally as the highlight head and the bottom of this slide, three case messages for the results. First, coating support addition from the proliferation of human bone marrow stromal cells for the 14 days, confirming ex situ compatibility. The second, there was a significant increase in the solubility to the 14 days in the coated scaffold. And finally, in the third, the ELP coating showed an initial increase in ALP gene expression under baseline condition, while suggests early osteogenic signals even without osteogenic media supplementation. The formulation challenge is optimizing the BPM2. And why? The BPM2 activity is really dependent on the chemical environment and concentration, and the manufacturing is important to an inactive or active this BPM2. And the method and the correct method is for the low concentration acidic environment, and the high concentrations the neutral environment, or the name maybe is PBS. This slide represents the result from the chlorioelectronic membrane assay, also known as the CME assay, which was used to evaluate the biocompatibility, the vascular integration, and the angiogenic support to the PCL and TME scaffolds. On the left side, we can see four microscopic photographs. The scaffold placed directly on the chlorioelectronic membrane of chicken bryon and embryonic to day 14. On the right side, we have the histological sections trained with two different staining methods. The table is organized by coating types in row and by staining methods in column. The first column uses alkaline blue and serious red staining, and the second column uses golden staining. Summary said at the bottom left of the slide, three critical conclusions that I can see. We can see the successful angiogenesis and the anatomical integration is correct, and finally, biocompatibility. Okay, this slide shows the histological contrast between the collagen spawns, this is a control, and the PCL-TME scaffolds. First, in the top, the control group is the collagen spawn with a BPM2. It shows a clear and dense black and red ring, confirmed by the golden trichrome, and a disturbing red staining, which tells us that the real mineralized bound tissue formed around it. Now, looking at the bottom, the PCL-TME scaffold shows the great tissue integrations around them. The tissue literally grows in the scaffold, but no formation was detected at all. So basically, the scaffold was really accepted by the body, but the subcutaneous environment just did not have the right cells to trigger bone formation. And this contrast is exactly what these use our conclusions. Okay, to do not delay the presentations, basically in this slide, I like to represent the implement size of the movie subcutaneous model. You can see here more detail, but because in the past slide is the same information, in this slide is more broken down. And the only difference is that these are the results in an animal. Okay, the conclusion of the presentation of the inform it's three. You can read the conclusions, but basically is the conclusion is enfocated to the three objectives of the inform, and in this past slides I to talk about different results because the ring is red, ring is black, because the circumstance which it is be compatibility or not on the graphics. But the conclusion is basically resume this information that I can see you in the past slides. And finally, our recommendations of this inform basically is simple. First, improve the in vivo part because the material evaluation a real bone environment. Now it's an animal or now it's subcutaneous only because it's to a bone and it's important to evaluation in a bone. The second is optimize the concentration of BPM dose. I can use speak in the past slides. The high concentration is bad to the experiment, but the low concentration is work better. But was it done systematically in the form. The third improve the mechanical properties, obviously, the best materials with some coating, etc. And finally, use a long-term study.